And I'm back. Let's talk about synapses. Neurons, those cells of the nervous system, must communicate with other cells, including other neurons, in order to carry out their functions. Again, this follows the cell-to-cell communication principle we talked about earlier in the semester. The area where the neuron meets the target cell is called a synapse. It is called a neuronal synapse if it's another neuron. And these can either be electrical synapses or chemical synapses. Neuronal synapses can occur between axon of one neuron and another part of another neuron, which shows here. We have the axon of this neuron interacting with either a dendrite or an axon of another one. Here we have axodendritic synapses, this is a dendrite. This is an axon, of one neuron, the dendrite of this one. So you have axodendritic between an axon of one neuron and a dendrite of another. You have axosomatic, which is between the axon of a neuron and a cell body of another. So here's entering the cell body. In words, anytime you see the word somatic, we're talking about cells. And then the axonic synapse is between the axon of one and the axon of another. So here's the axodendritic, here's the axosomatic, and here's the axonic. Terms are used to describe which neuron is sending and which is receiving. You have a presynaptic neuron, which is the neuron that is sending the message from its axon terminals. And you have the postsynaptic neuron, which is the one that is receiving the message. Synaptic transmission can be chemicals or electrical. The signals can be chemical or electrical between the neurons at the synapse. These are the fundamental process for most functions in the nervous system. These allow for voluntary movement, cognition, sensation, and emotion. The average presynaptic neuron forms synapses with about 1,000 postsynaptic neurons, and the postsynaptic neuron can have as many as 10,000 synaptic connections with different presynaptic neurons. The electrical synapse occurs between cells electrically coupled via gap junctions, which we will see in just a moment. The axolemmas of one of each cell in the synapse are generally touching. The gap junctions align channels forming pores that ions and other small substances can flow through. In certain areas of the brain responsible for programmed or automatic behaviors such as breathing, you will find electrical synapses, and you will find electrical synapses in cardiac and visceral smooth muscle to allow for coordinated muscle activities. The electrical current can flow directly from the axoplasm of one neuron to the next. Creates two unique features of electrical synapses. That is, the transmission is bidirectional, which means it can be pre or post synaptic. Either neuron can be pre or post, depending on the direction that the current flows between them. And transmission is nearly instantaneous. There's only a delay in time for the presynaptic neuron to depolarize. This is less than 0.1 millisecond. So that's 1/10 of a millisecond. These are much faster than chemical synapses, which are 1 or more milliseconds. And there is the electrical synapse. You see the axon terminal of one, the axon of the other, and they are literally touching. There are your gap junctions forming these bridges or pores where ions are free to travel from one side to the next. A chemical synapse, this is the type that makes up the majority of synapses in the nervous system. They are more efficient than electrical synapses. They convert electrical signals into chemical signals. Therefore, no signal strength is lost. We do lose in electrical synapses. There is energy that is lost. In a comparison, you have there are structural differences. In a synaptic or in a chemical synapse, you're going to have synaptic vesicles. These are filled with neurotransmitters, the chemical messengers, transmits the signals from the presynaptic to postsynaptic in one direction only. The synaptic cleft is going to be a small ECF, always stands for extracellular fluid space, separates the presynaptic and postsynaptic neurons. In these chemical synaptics, the gap junctions connect neurons in the electrical synapses. So if you remember, here we have these gap junctions that are holding things together. Those are absent in the chemical synapse. The postsynaptic neuron has neurotransmitter receptors, which bind to the neurotransmitters which are secreted from the presynaptic neuron. The time gap between the arrival of the action potential at the axon terminal and the effect of the postsynaptic membrane is called the synaptic delay. Again, chemical synapses are unidirectional. They only go one way, which allows for a variable signal intensities. The more neurotransmitter that is released from the presynaptic neuron leads to a stronger response at the postsynaptic neuron. And here is a typical chemical synapse. There's the axon terminal. There's the effector cell. There are the neurotransmitter receptors. Here are the neurotransmitters. Those neurotransmitters will be released. They will bind to the receptor, which then sends the signal on to either the effector organ or to the next axon. The things that happen at a chemical synapse are much more complicated than neuromuscular junctions. There are multiple neurons secreting many different types of excitatory or inhibitory transmitters. The action potential in a presynaptic neuron triggers opening of voltage-gated calcium ion channels. The influx of calcium causes synaptic vesicles to release neurotransmitters. The neurotransmitters then bind to the receptors on the postsynaptic neuron. This causes ion channels to open, leading to a local potential and possibly an action potential if the threshold is reached. So here again, here's the action potential in the presynaptic, triggers the opening of the calcium. Calcium coming in is what triggers the release of the neurotransmitters. The neurotransmitters then exit into the synaptic cleft, where they are picked up by the postsynaptic neurons and release the neurons into the next, either the effector organ or the next neuron. Postsynaptic potentials are local potentials in the membranes of the postsomatic neuron. That's what's going on once the neurotransmitters are released. Neurotransmitters here opens and allows for the movement of ions. The membrane potential of the postsynaptic neuron moves closer to the threshold, caused by local depolarization. This is called the excitatory postsynaptic potential. Membrane potential of the postsynaptic neuron moves further away from the threshold, caused by local hyperpolarization. This is called inhibitory postsynaptic potential. You see the dip in the potential. Here's the threshold. As we are moving toward the threshold, that is when the signal will be sent forward. Once that threshold is reached, if that threshold is never reached, the signal stops at this particular point. Neurons receive input, both inhibitory and excitatory, from multiple neurons, each of which influences whether the potential is generated. Neural integration is the process in which the postsynaptic neuron integrates all incoming information into a single effect. Summation, again, anytime you see the word summation, that means putting everything together. All the input from several postsynaptic terminal potentials are added together to affect membrane potential at a trigger zone. The action potential will only be generated if the threshold is reached. The sum of the EPSPs must be enough to overcome the sum of the IPSPs. If the sum of the IPSPs is greater than the EPSPs, the membrane will hyperpolarize, threshold will not be reached, and an action potential will not be generated. There are two different types of summation. Temporal, this is where the neurotransmitter is released repeatedly from the axon terminal of a single presynaptic neuron. Each local potential is short-lived. It must be generated quickly enough to reach the threshold and create the action potential. Spatial summation, stimulus release of neurotransmitters from the axon terminal of many presynaptic neurons. IPSPs are also subject to both temporal and spatial summation, but have inhibitory effects which make it less likely to reach the threshold with subsequent action potential generation. So here is a membrane potential trigger zone. We have the first EPSP, the second EPSP, the third EPSP. We've reached potential, and action potential will be created. Here's the stimulus. All of them comes and we reach our threshold. So this is sort of a stair step, and this is more of a ramp. Synaptic transmission may be terminated by ending its effects of neurotransmitter. The neurotransmitters diffuse away from the synaptic cleft in the extracellular fluid, can be reabsorbed into the neuron or the astrocyte. The neurotransmitters may be broken down in the synaptic cleft by enzymes, the byproducts of which will then be resorbed by the presynaptic membrane for reassembly. And some neurotransmitters are reabsorbed in the presynaptic neuron in a process called reuptake. So there is the chemical synapses showing this one is being taken up. This is reuptake, where the neurotransmitter is simply brought back. Here's where the degradation occurs, or here the neurotransmitter is simply diffusing away and being absorbed into the body's immune system, or by the body's immune system, and being removed by the blood and other processes that remove cellular waste. Here the neurotransmitter is being degraded, and all of these little parts will be resorbed back into the presynaptic neuron and reassembled. Distribution of protein channels and pumps is predictable if functions are considered. Ligand-gated ion channels bind neurotransmitters from another neuron. So ligand-gated ion channels will be located on receptive regions of the neuron, the dendrites in the cell body. Voltage-gated sodium-potassium ion channels, which open and close during action potentials to send the signals, are generally going to be located on the part of the neuron that sends the signals to other cells, such as the axon. The voltage-gated calcium channels, which trigger exocytosis of the synaptic vessels, only one place in the neuron, and that's where the synaptic vessels are located, which is the axon terminal. And the leak channels for sodium-potassium pumps generate and maintain resting membrane potential. That resting membrane potential applies to the entire neuron, so leak channels and sodium-potassium pumps are located throughout every part of the neuron's membrane. So you're going to have leak channels all up and down, voltage calcium channels here, voltage potassium channels here, and voltage-gated sodium channels here. And then the ligand-gated channels on this end of the dendrites. Venomous arthropods in the United States, including spiders and scorpions. Many venoms actually affect neuronal synapses. These are called neurotoxins. The black widow spider, which we have here in Arkansas, I kill one or two of them in my garage every year. I pay a man to come around and spray my house and spray my yard, but I always manage to have a few black widow spiders every year. The toxin causes a massive release of neurotransmitters, which causes repetitive stimulation of the postsynaptic neurons. The black scorpion is the most lethal of the 40 species of scorpions in the United States. Its venom prevents postsynaptic sodium channels from closing. The membrane remains polarized and continues to fire action potential. Either one of these common symptoms are muscular hyperexcitability, sweating, nausea, and vomiting. And due to the effect they have on the nervous system, They affect the breathing muscles, and you have difficulty breathing. Depending on the amount of venom received and the availability of medical care, severe cases usually require antivenom to block the effects of the toxin. Again, treatment and prognosis based on how much did the scorpion or the spider inject in you, how close is medical care, how quickly can you get to medical care, etc. This is the overall big picture of the chemical synapse showing the synaptic vesicles here, the neurotransmitters, all the uptake processes, etc. Neurotransmitters. Nearly all neurotransmitters undergo a similar pattern of use despite where or that there are over 100 known. They share some very similar features. They are made in the cell body of the axon terminal and packaged in structures called synaptic vesicles. They are released from the axon terminals of the presynaptic neurons. They cross the synaptic cleft. They bind to very specific receptors on the postsynaptic membrane, and their effects are often rapidly terminated through removal and/or degradation. There are several different types of neurotransmitters that are receptors on the postsynaptic membranes. Ionotropic receptors are a component of ligand-gated ion channels. They can directly control movement of ions into and out of the neuron when they bind to the neurotransmitters. There's metabotropic receptors within the plasma membrane. They're associated with separate ion channels. They are connected to metabolic processes and initiated when the neurotransmitter binds. There are G proteins, which are a group of intercellular enzymes associated with many metabotropic receptors. They activate a cascade of enzyme-catalyzed reactions from the intercellular chemical messenger molecules called second messengers. The neurotransmitter is, of course, the first messenger. Second messengers open and close ion channels in the postsynaptic membranes. Cyclic adenosine monophosphate, or CAMP or CAMP, is a common second messenger derived from ATP. It has multiple functions in neurons. Remember, we talked about adenosine diphosphate and adenosine triphosphate. Give me just a second. Thank you. I had to get a swallow of something. Throat's getting scratchy. This is adenosine monophosphate. Only has a single phosphate. Again, cells can derive it from ATP. Remove two of the phosphates. CAMP binds to a group of enzymes, which adds phosphate groups to ion channels and triggers the channels to open and close. So here's the ionotropic receptor when it's closed. There's the binding site for the neurotransmitter. Neurotransmitter binds, channel opens, allows the ions to move back and forth. Metabotropic receptors, the ion channel is closed. The G protein, which is inactive, is bound here. The neurotransmitter binds here, causes the G protein to become active. The enzyme reacts and sends out the second messengers. Second messengers come over and open the ion channel. Binding of neurotransmitters to the receptor leads to an EPSP excitatory effects or an IPSP inhibitory effect. Most neurotransmitters can have both effects depending on which postsynaptic neuron receptors they bind to. Single neurotransmitters may have several receptor types, and major neurotransmitters are classified into 4 groups based on their chemical structure. And here is a chart showing the most common neurotransmitters and neurotransmitter types, their precursor compounds, their postsynaptic effect, their predominant locations, and the types of receptors. Again, acetylcholine is one we always talk about. Acetylcoenzyme A and choline. It is an excitatory found in the central nervous system, in the brain and spinal cord, peripheral nervous system, at the neuromuscular junctions in the autonomic nervous system, and it has two types of receptors. And then there are lots of others. There's histamine, serotonin. Most of us have probably heard about that one. There's histamine. Everybody's heard about that one. Glycine, opioids, neurotransmitters. Lots of talk about opioids. The opioid drugs are going to affect these neurotransmitters. Again, major neurotransmitters for brain control. Acetylcholine. Acetylcholine is a small molecule neurotransmitter widely used by the nervous system. Cholinergic synapses bind acetylcholine in neuromuscular junctions within the brain and spinal cord and within the autonomic nervous system. They are largely excitatory, but they do exhibit some inhibitory effects in the peripheral nervous system. They are synthesized from choline and acetylcoenzyme A. Remember, acetylcoenzyme A is one of those things we find in cellular respiration. It is impacted in synaptic vesicles. It is quickly degraded by acetylcholinesterase, an enzyme in synaptic cleft. Byproducts are taken back into the presynaptic neuron for recycling and reuse. The biogenic amines, or the monoamines, are 5 neurotransmitters synthesized from amino acids. They are used throughout the central nervous system and peripheral nervous system for regulation of homeostasis and cognition. The first 3 form the cathephylamine group made from the amino acid tyrosine. They are mostly excitatory. You have probably heard of norepinephrine. It's mainly found in the autonomic nervous system. It influences heart rate, blood pressure, and digestion. In the central nervous system, it regulates the sleep-wake cycle, attention, and feeding behaviors. Epinephrine, also found in the autonomic nervous system, has similar functions as norepinephrine. It is more widely used as a hormone by the endocrine system. Dopamine, used extensively by the central nervous system, involved in movement and coordination, emotion, and motivation. There was a movie made back in the 80s or 90s. Robin Williams played a doctor called Patch Adams, who did some work with a synthetic form of dopamine. The movie was called Awakenings. It's actually a pretty good movie. If you have a rainy Sunday with a couple hours to kill, I'm sure you can find it on Netflix or Hulu or somewhere. Serotonin is synthesized from the amino acid tryptophan. It's the most serotonin secreting neurons are in the brainstem. Axon project into multiple areas of the brain. Functions include mood regulation, emotions, attentions, feeding, and daily rhythms. Histamine is synthesized from the amino acid histidine, which has a regulation of arousal and attention. Glutamate, the most important excitatory neurotransmitter in the central nervous system, binds with the ionotropic postsynaptic receptors, opens channels that will allow flow of both sodium and calcium ions, and generates EPSPs in the postsynaptic neuron. And then glycine and GABA are both major inhibitory neurotransmitters. They induce IPSPs on the postsynaptic neuron by opening chloride channels and hyperpolarizing the axolemma. The neuropeptides are a group of neurotransmitters with wide variety of functions. They must be synthesized in the cell body and transported to the axon. Substance P is released from type C sensory afferent vesicles that carry information about pain and temperature. Think about P and pain. Also released by other neurons in the brain, spinal cord, and gut. The opioids are a group of more than 20 neuropeptides, including the endorphins, the enkephalines, and the dimorphins. All elicit pain relief. They are nervous system depressants. And neuropeptide Y controls feeding behavior, may mediate hunger and the feeling of being full. Certain psychiatric disorders and treatments are the result of neurotransmitters gone wrong. But psychiatric disorders affect thought processes. They are generally treated by modifying synaptic transmission to change how neurons communicate. The study of drugs that affect higher brain function is called psychopharmacology. These drugs target either the action potential generation or some aspect of the neurotransmitter physiology. Schizophrenia is repetitive psychotic episodes. These are episodes during which the patient is unable to appropriately test beliefs and perceptions against reality. Folks with schizophrenia think there's somebody in the walls trying to get at them. There's bugs under their skin, all types of things going on. It's thought to be the result of an excess release of dopamine. Management involves blocking those postsynaptic dopamine receptors. Depressive disorders, lots of talk about depression in the United States and around the world the last year, with people being closed in, can't go to movies, can't go to theater. For a long time we couldn't go to the grocery store, we couldn't go anywhere, and depression was on the rise. But depression is defined as simply disturbances in mood. These are thought to be the result of deficiency in the synaptic transmission of serotonin, norepinephrine, and/or dopamine. The most widely used antidepressants are selective serotonin reuptake inhibitors. These block serotonin transporter and prevent the reuptake by the presynaptic vesicles. Anxiety disorders are those that are exaggerated and inappropriate fear responses. Believed to stem from abnormalities in norepinephrine, serotonin, and GABA transmission. They're treated with antidepressants, GABA activity enhancers, and others that modulate norepinephrine transmission. Bipolar disorder, been a lot in the news lately about bipolar disorder. Bipolar disorder is characterized by episodes of an abnormally elevated mood called mania, followed by a severe depression. People who suffer from bipolar disorder, for instance, will be laughing 1 minute and for no particular reason crying hysterically 2 minutes later. Treatment involves decreasing the ease of action potential generation, generally blocking sodium channels in the axonal. All right, I'm going to stop there and I will pick up with the functional groups of neurons and probably finish this chapter the next time.